Chapter 8. So in Chapter 8, we're going to be talking about microbial genetics. So this one we're going to talk about DNA replication, and gene expression again. Some of you have already done gene expression courses like General Biology 1 or Anatomy and Physiology 1. That'll be a review for you. But for me, some of you, unless you've had General Biology 1, DNA replication will be a new concept. I chose the quote that opportunity does not knock. It presents itself when you beat down the door. So anyway, as we get started, this is what we'll be talking about. There'll be some extra stuff that we talk about that goes beyond what we do in a biology class. It's specific to bacteria, things like conjugation and things like that. So anyway, we're going to get started when we talk about genetics, and that is really just the study of genes, study of how we get our traits, the study of how what DNA says becomes what you have. Okay, the study of what genes are, how they carry information, how that is expressed, and how they're replicated. Okay, so basically it's the scientific study of genetic information. Genomes, that is, a genome is really all of a cell's DNA, all the genetic information in a cell. You have a genome, and bacteria have a genome. Viruses have a genome, things like that, okay? Genomics is the study of that. Study of genomes. When we molecularly sequence the genome, we want to understand what every gene is and what it does. When we completed the Human Genome Project, that wasn't actually fully complete. There was still more to do. We recently actually finished the Human Genome, not that long ago. And I always forget exactly, but genomics. Now, one of the things is we have chromosomes. That is a structure that holds DNA. Physically, DNA is in a structure called a chromosome. Chromosomes are different if you look at bacteria versus, so you look at prokaryotic chromosomes versus eukaryotic chromosomes. They're very different, okay? A gene is just nothing more than a bit of DNA that codes for a protein. It's a segment DNA that actually codes for a functional polypeptide. It could be an enzyme, but it's going to be some functional product. It's going to be a protein, okay? Like an enzyme, something like that, okay? All right, now, genetic code is a set of rules that tell us how to read DNA, okay? How they determine what nucleotide sequence, what does that actually become? What amino acid sequence does it become? What polypeptide does it become? Genotype, that is the actual combination of genes that we have. Okay. Well, I frequently call for us, for eukaryotes, I always say it's your pair of alleles. Genotype is what the gene says. It's what the DNA says. Phenotype, that's what the protein says. That's the physical expression of a gene. That's the polypeptide. That's the appearance. That's the actual biochemical end product. Another way to say phenotype is polypeptide or protein. Okay? A plasmid is some extra DNA. It's a small circular DNA molecule. Replicates separately, independently of your plasmid. Okay, of your other chromosome rather. So your plasmid is its own little entity. So one other thing we do, we start with our bacterial chromosome. Now bacterial chromosomes there's one of them usually, and they are circular. That DNA will be transcribed into messenger RNA, and that messenger RNA will be translated into a polypeptide. And that is the basic genetic flow of gene expression. In bacterial recombination, we can add, transfer stuff and information between cells of the same generation. And then in replication, we are going to copy our DNA so that our two cells can separate. Okay? So that's the basic genetic information. We have expression, recombination, and replication. Okay? Now, DNA double helix. What is this? DNA is a double helix. Now, one of the things I like to do when I teach this is I like to, I said that if I had a pentose sugar here, okay, there's 1, 2, 3, 4, 5 carbons in a pentose, okay? This is a pentose, okay? Now there's 5 carbons. We can number them starting from this oxygen and going clockwise, okay? We have our 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime carbons. Notice that this end here is sticking down and this end is sticking up. Okay? So there is a 5 prime end and a 3 prime end. Okay? What this will do is cause our DNA molecule that we have, okay, our DNA molecule to have a 5 prime and a 3 prime end sticking up like this. But what happens is on the other end it's the opposite, okay? So what you have is this anti-parallel nature of DNA. Okay, we call this anti-parallel. The double helix has a twist held together by hydrogen bonds. Carbon atoms 1 to 5 on the pentose. 5 prime having a phosphate. 1 strand is 5 to 3, and the complementary is 3 to 5. Okay. All right. Now, see, I'm gonna sneeze. Excuse me. Now, DNA is a double helix. There's a sugar phosphate backbone. There's a 5 carbon sugar. In DNA, it's deoxyribose. In RNA, it's actually ribose. There's a phosphate group. Okay. There's nitrogenous bases. There are 2 groups, purines and pyrimidines. If you're pure, you're all good. Adenine, guanine, or purines. And you cut pyrimidines are cytosine, uracil, and thymine. Remember that in DNA, there is only going to be thymine, and in RNA, there's going to be uracil. Uracil is found in RNA only, thymine is found in DNA only. Okay? Alright. Now that's about DNA replication. Now, DNA replication can be, at first blush, hard to understand. What I want to talk about is that there's going to be 2 complementary strands of DNA that a normal DNA has. It's going to create daughter strands that are identical to them, okay, to the original DNA. This is what makes your sister chromatids in your DNA's DNA replication. DNA is semi-conservative, and we're going to talk about what that means. So let me explain it, then I'll talk about it, okay? Now, let's say I have a DNA molecule here, okay? We have this DNA molecule that we have, okay? And this DNA molecule is all old DNA. It's an old DNA. We got a cell. This cell is getting ready to undergo binary fission or mitosis, okay? It could be a bacterial cell getting ready to undergo binary fission. It could be a eukaryotic cell getting ready to do mitosis. What's going to happen is we're going to take this DNA and we are going to split this in half. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. We're going to split this DNA in half. Then what we're going to do is we're going to make some DNA that is identical to it, but in terms of the same complementary strands identical to our parent. Okay, so what's going to happen here is you're going to be half new, half old. Okay, so blue being new and black being old. Okay, this is called semi-conservative replication. Okay, so it's semi-conservative. It isn't all old. It isn't all new. It's half new, half old. Okay. Now, basically, what's going to happen here is this. Okay. Let me explain it a different way. Let's say I had a DNA sequence here. Cat gag. I always joke that's the DNA sequence that causes hairballs in cats. Is cat gag. Okay. Then C will bind to G, A will bind to T, T will bind to A, G to C, A to T, and G to C. And we have our complementary strand. This cell will now get ready to divide, and we got to divide up our DNA. We'll get cat gag over here, and we'll get GTA, CTC over here. Okay? And what we've got to do is we've got to pair these to new stuff. GTA, CTC, and cat gag. Okay, so what you have is two strands that are identical. Okay, they are the same as this strand, but now there's twice as much of it, but it is half old and half new. Okay, I hope that made sense. This is what we mean by semi-conservative DNA replication. Now, what I'm going to do is I'm going to draw this out and talk about it. I want to make sure that I don't leave anything out. What I was going to do is just, okay, good. I talk about all the enzymes that I think I should talk about. Alright, so what I'm going to do for DNA replication is I'm just going to teach it, then I'm going to talk about it. Okay, this is how I like to do things. So what we're going to do, so keep in mind DNA replicates semi-conservatively, okay? Now, so I've got a DNA strand right here. Now this black is going to be our old DNA, okay? We have let's add a 5' and 3' in here. Let's add a 3' and 5' in there. Right here we have what is called an origin of replication. Now this origin site, what's going to happen is the DNA's molecular machinery is going to come in and pry this apart and create what I like to call a replication bubble. It makes a bubble. Now, one of the ways it does this, there's this origin site, and there's a chemical, an enzyme that comes in, and this enzyme kind of acts like a DNA zipper, okay? And I'm just going to write H on there. This enzyme's name is helicase, okay? One of my favorite biology pickup lines is, girl, if I was an enzyme, I'd be helicase so I can unzip your genes. It unwinds and unzips your DNA, okay? Unzips your genes, your genetic material, okay? What it's doing is going this way and that way, and it's making a bubble, and it's prying the DNA apart and making what is called a replication fork. Okay, a replication fork. There's one over here as well. There's a replication fork. Okay, these replication forks that we're making We're unwinding and unzipping our DNA. We're making a fork. They're going both directions away from the origin of replication. What we've now got to do is, you remember my black? I built my black and I had my all that going on. I built my blue on top of that. We're going to do that in a minute. Now, so this doesn't twist. When this is unwinding, we got something that's going ahead of all this, an enzyme that's going ahead. Now, very important we talk about it because this can be a way that antibiotics can prohibit these from working. Is there's an enzyme called topoisomerase. Okay. Topoisomerase basically relaxes the DNA in front of the helicase so that you don't shred your DNA. Okay? Now, what we've got to do is inside here, we're going to, so I like to think of this as like, let's say you're laying a tile floor, and the first tile you lay is the most important tile because that's going to dictate where all the other tiles go. So laying that first tile is kind of important. It's the one it's a really important tile to lay down. Okay, so we're going to lay our first tile, and our first tile that we're going to lay down is our building block to build everything off of. Okay, what this is a little primer made of RNA. It's an RNA primer. All of these are RNA primers. These RNA primers, what they are going to be is they're enzymes that build DNA. And the enzymes that build DNA, they do this purely because they can only add to the 3' end of nucleotides, because that's where the OH is on DNA. That's where a dehydration synthesis can occur. So that 3' end is the only one that's free. So what happens is we have a 3' end and a 5' end. Okay? And we have a 5' and, sorry, we have a 3' and a 5' there. Okay? Now on this one over here, we have a 5' and 3'. And we have a 5' and a 3'. Now we can only add to the 3' end. Okay? And this is how the DNA is going to work. This is the ordering. Notice this is 3, this is 5, this is 3, this is 5. This that we're making is going to be the same as this, and this that we're making is going to be the same as that. And we're going to make 2 DNA strands that are identical, just like in my drawing with the letters of the nitrogenous bases and my drawing with just lines. We're just showing you how it mechanistically happens. Now, DNA can only add to the 3' end. So what's going to happen is you got a 3' end right here. DNA replication can only go from 5 to 3' direction. So what's going to happen is that this 3' end being free, we're going to be able to grow DNA from it straight towards the fork. The fork's moving this way, it's moving, and this is chugging behind it. Just chugging along, falling along behind it. Okay? Now, what's going to happen here is, on this one, we can't, because we can go this way. Okay? We could do that, but that's as far as we can go. So we have a problem on this side. To fix that problem, we're going to come in here behind it with another little piece of RNA primer. Okay? And what we're going to do is we're going to build our DNA like this. We'll build a little piece. Well, we got the same problem. Let's go right here behind that. Okay? And let's add a little piece. Okay? So what happens is there was no 3' end here. We needed a 3' end to build this off. We needed a 3' end right here. So we have to come in with another primer, and we got to use several primers and build DNA in little pieces and build it away from the fork. Here, because there is a 3' end, we can build DNA continuously towards the fork. Now if we look over here, we got the same problem. There's a 5' end here, and there's a 3' end here. It flips. So we can build that one continuously. Over here, I've got to come over. I've got to build a little piece, okay, and build it away from the fork in little pieces, okay. I have to build it in little pieces away from the fork, okay. You have two rules that are important. You can only add to the 3' end and go in the 5 to 3 direction, okay. Now, this line right here, this strand, also that one, is called the leading strand, okay. Think about it. If you were in a race and the replication fork was the finish line and you run continuously towards the finish line, you'll probably be in the lead, especially if the person you're running against is going the wrong way and they're going in pieces. This strand is called the lagging strand, okay. So is that one, the lagging strand, okay. All right. Now, so that the DNA, when we pull it apart so it don't bind to itself, frequently what we'll do with it is we'll stick some proteins on it so it don't bind to itself. What we in biochemistry call hairpinning. We'll stick these on there. These things are called single-stranded binding proteins. Okay? Single-stranded binding proteins. Okay? The SSBP, the single-stranded binding protein, they're going to prevent what we call hairpinning. Okay? All right. So the replication point is where replication is happening. There's origins of replication that I'll talk about here in just a minute. The replication fork, we have semi-conservative, where each double-stranded DNA molecule has one original, one new. The fork moves across the parental DNA as it unwinds. The single strand combines with new, making a newly made strand and original strand. They will separate, they will rewind and become new DNA strands. The leading strand is replicated continuously. It's made by an enzyme called DNA polymerase 3. Okay? DNA polymerase 3, and lagging strand can be synthesized discontinuously. Now, leading strand is made continuously towards the fork. Lagging strand is made discontinuously and in fragments away from the fork, and that'll be something that I'll test on when we get there. There's RNA primers that must be created first, and by the way, I forgot to mention this in this part. These little bits that we were talking about right here, these little things here, they are called Okazaki fragments. Okay. All right. That short segment is a single-strand DNA formed in the lagging strand. Okay. All right. Now, DNA polymerase 3 cannot add to the 5' end. The DNA polymerases can only add to the 3' end. Okay. So what happens? We have our origins of replication. Replication begins at special sites called origins of replication, where the two DNA separate and forms what I like to call a replication bubble. The eukaryotic chromosomes, they have many thousands of origins of replication sites. Prokaryotic chromosomes only have one. So in a prokaryotic chromosome is a circle. There is one origin. It starts replicating like this and separates our two DNA. Okay, that's how it works. Okay, and we're going to focus on that because this is DNA of bacteria. This is a microclass. The enzymes that link nucleotides can only add to the 3' end, never the 5' end, and DNA can only grow to 5' 3' direction. That's very important to keep straight. Okay. Now, the replication fork is a Y-shaped area where the enzyme helicase is moving down, unwinding and unzipping our DNA. It unwinds and untwists the DNA double helix. It literally unzips your genes. Okay? There are single-stranded binding proteins, the SSBPs. Here you can actually see them here and here. Those single-stranded binding proteins stabilize a single strand so it can be used as a template. It prevents DNA to bind to itself. We call that DNA hairpinning. Gyrase is one enzyme that prevents overwinding. Topoisomerase is the big one that I talk about. It prevents overwinding. It prevents the DNA from winding itself too much, where it relaxes ahead of the fork, relaxes it so that it is unwound and unzipped so it doesn't twist itself and shear. Okay. Primers are added. The primers, these are going to be building blocks that we add, small RNA nucleotide areas that we add, small bits of RNA that we add that serves as a 3' end to build off our DNA. An enzyme called primase will actually start that, add that RNA to it, and then what we'll do is when we're done, the primer will then be a building block to add DNA off of it. Okay, our book doesn't do a good job of showing that. That's why I did the drawing. I don't think the book does a great job of showing how DNA replication happens in a good diagram. Polymerases, DNA polymerases, which there's multiple ones, they catalyze making the new DNA. The antiparallel structure of your double helix is going to affect the two strands differently. Now they can only grow 3 to 5. DNA can only elongate in 5. They can only add to the 3' end and only grow in the 5 to 3 direction. There we go. Okay, two strands oriented in opposite directions. One strand, the 3'5 strand, is replicated continuously towards a fork called the leading strand. Think of the replication fork as your finish line of a race. You have two runners. One runner's running continuously towards the finish line. They're going to be in the lead. The runner who's running away from the finish line and in some running, he will running backwards, running the wrong direction, and running in fragments, will be in the lagging behind. Okay. DNA polymerase 3 is responsible for making this strand. The lagging strand is the 5' strand. It is DNA polymerase must replicate away from the fork in pieces. These fragments are called Okazaki fragments. DNA ligase links all the primers together, and it's synthesized by DNA polymerase 1 and 3. DNA polymerase replaces the RNA nucleotides of the primer. So now the primer was there. It is now DNA. Ligase will come in and link all this together. It takes all the primers and Okazaki fragments and links it together. Now the lagging strand is complete. The leading strand has a little bit of that's done. The DNA polymerase 3 adds nucleotides to your leading strand. Lagging strand uses DNA polymerase 1 and 3. RNA polymerase add many nucleotides. There are many RNA primers found in the lagging strand, where there's only one found in the leading strand on one side. Okay, RNA polymerase 3 adds DNA to the RNA primer. DNA polymerase 1 does replace the DNA nucleotides. It doesn't really build as much as it does the DNA polymerase. More checks itself before it wrecks itself. DNA ligase joins and links the fragments together. And then you're done with replication. Bacterial chromosomes are circle. They go bidirectionally from the origin around like this. Okay, till you make it. This is actually the very first video ever of DNA replication. Sorry, So this is actually the very first microscopic footage of DNA actually replicating itself. Showing this was actually the first ever video of that. So anyway, I wanted to show you that. Alright guys, I'm going to stop here and pick up in the next one.